Bismillahirrahmanirrahim. Today the topic of my discussion is inhibitors of electron transport chain and oxidative phosphorylation and uncouplers. Inhibitors, these include inhibitors of complex 1, complex 2, complex 3 and complex 4 and inhibitors of oxidative phosphorylation. As I have told you in previous lectures of electron transport chain that there are total 5 complexes, out of which 4 are complexes and the fifth one is the ATP synthase. The 4 complexes are responsible for the transport of electrons along the electron transport chain. So every complex have its own inhibitors which inhibit the electron transport chain. We will discuss one by one the inhibitors of 4 complexes. First of all, the inhibitors of complex 1, pyridine, guanethidine, rotenone, barbiturates. These are, rotenone is the fish poison, barbiturates are antidepressant. Secondly, inhibitors of complex 2, TTFA, that is the iron chelating agent, malonate, which inhibit the succinate dehydrogenase, and then is the carboxyl. Then are the inhibitors of complex 3, antimycin, British and Leviside, BL, fenformin. Then are the inhibitors of complex 4, carbon monoxide, azide cyanide, that is represented as Cn-1, and hydrogen sulfide, that is represented by H2S, all gases that inhibits the complex 4 of the cellular respiration. These are all gases which inhibit the electron transport chain which are present in the complex 4. So complex 4 have the inhibitors that are all gases, that are the carbon monoxide, azide, cyanide and hydrogen sulfides. Inhibitors of an oxidative phosphorylation, that includes the acetyloside, inhibits the translocation of ADP and inorganic phosphate, oligomycin. Various potassium ionophores are there, which includes the varenomycin and gramicidin. Ionophores disturb the proton gradient across the mitochondrial membrane. So these are the inhibitors of oxidative phosphorylation. Next is the uncouplers. There are two types of uncouplers, physiological and chemical. First of all, we will discuss about the mechanism of uncoupler action. The mechanism of uncouplers action is they increase the intermitochondrial membrane permeability. The uncouplers increase the intermitochondrial membrane permeability. How? As we know that the membrane is impermeable to protons. So there is a development of proton gradient. Once it is permeable to proton, there is no development of proton gradient through the membrane and so no senses of ATP. So it makes the intermitochondrial membrane permeable. So once the membrane is permeable, it does not produce ATP. These uncouplers dealing with the oxidative phosphorylation from the electron transport chain, so that's why there is only generation of heat. So there is no production of ATP, but they produce heat. Physiological uncouplers include thyroxine, long chain fatty acids, unconjugated bilirubin, thermogenin. What is thermogenin? Brown adipose tissue, mainly it is present in the newborn and it is present in the hibernating animals which hibernate themselves during the winter season. It is also responsible for the generation of heat without ATP, that's why it is important. Chemical uncouplers include 2,4-dinitrophenol, 2,4-dinitroglycerol. So these are the two substances that are responsible for delinking of electron transport chain from oxidative phosphorylation. What is oxidative phosphorylation? As we know that in oxidative phosphorylation ATP is produced and the electron transport chain occurs and oxidative phosphorylation in ATP is produced, but the uncouplers inhibit the production of ATP, but they produce heat without the formation of ATP. Followed by these two there is the pentachlorophenol, FCCP, that stands for fluorocarbonyl cyanide phenyl hydrazine, aspirin hydroses and calciamine Cu2 positive and dicumarol. These are the chemical uncouplers that are responsible for the generation of heat without ATP production. So what are uncouplers? Those chemical substances, some are physiological and some are chemical, which are responsible for the production of heat without the production of ATP, because they make the membrane of the mitochondria permeable to the hydrogen ions. So when the membrane become permeable, then it does not causes the membrane potential. So when there is no membrane potential, there is no production of ATP, and so these uncouplers are responsible for the production of heat without the ATP production. So that was all about the uncouplers and the inhibitors of ETC, that is electron transport chain oxidative phosphorylation. So there is a short note in the examination: what are the inhibitors of electron transport chain and oxidative phosphorylation? And very important short question, that is what are uncouplers and what are their types, and define uncouplers, what are their types, write a short note on uncouplers. So that was all about the uncouplers and inhibitors of electron transport chain. Thank you very much for watching. If you have any query related to this topic, then you can ask in the comment section.